chapter 4 is biological molecules. Biological molecules. So without wasting time, let's look at biological molecules. What are the things we're expected to know here in this chapter? One of the first things we need to know is to know about water. This section in chapter 4, part 1, we're going to look at the need for food. Why do we need food? You know, water falls in the category of food. Okay, water is part of food. We're looking at the dietary, the nutrients. Water is part of the group. So we're looking at the first one, which is water. Now, water is the only of the biological molecules that do not have the carbon. You know, water do not contain the carbon. The carbohydrate, fat, protein, the rest of them, they contain carbon. They contain carbon. Water do not have carbon in it. It just have hydrogen and oxygen in ratio 2:1. Okay? Now, let's quickly take a look at why do animals need water? Why do plants, animals need water? First of all, the benefit of water to organisms. Number one is that water is used as a medium for chemical reactions. Water enables chemical reactions to take place. Without water, reaction cannot take place. Like you bring this, you bring that, you mix them together, they cannot react, you know. Unless you add water, you put water, the presence of water, then reaction will easily take place. So it's a medium for chemical reactions to take place, number one. Number two, another function of water in animal, we're looking at it from the animal perspective, we look at it also from the plant's perspective. There's a second function water do to animal body, to human being. You know, human being belongs to the category of animals. So another thing water does in our body is that water helps to transport the digested food. Okay, all the digested food products like your carbohydrate, the digested carbohydrate, the digested glucose, the digested fat, digested protein, all of them are transported with water, with the help of water. They mix with water and they move around the body, around the entire body. So that the transport of digested food is done with the water. So not only the digested food product, also the secretory product like the hormones. Hormones do not move on their own, cannot move on their own if they are not carried in water. Hormones, blood or water, you know, blood is mostly liquid, mostly water. The liquid part of the blood is called the plasma. Okay, we're going to talk about that later on. So water is, the blood is systematically made up of water. So water helps to carry hormones. Remember, these hormones are carried in blood and they are secreted or they are deposited wherever they are needed in every cells that needs in the body to be deposited to that cell. So it is water that helps to carry both digested food, helps to carry secretory products like hormones, okay, from one part of the body to another part. That's a second function of water in animal. Another function of water in animal is that it's very essential part of the cytoplasm. When you look at the animal cell, the cytoplasm is largely made up of water. Cytoplasm is largely made up of water. So water is an essential part of the cytoplasm. Not only cytoplasm, it's also an essential part of the lubricants, like the mucus that helps to grease the parts. Okay, water is largely part of the lubricant. Water is also largely part of the digestive juices in the body, in the stomach. Both the gastric juices in the stomach and every other juice and in the intestine, small intestine, water is largely made up of all those juices. So water is largely made up of cytoplasm, lubricant, and digestive juices, and even the blood. Water is largely made up of the blood in our body. Now, that's the third functionality of water. Now, the fourth in animals. Now, the fourth function of water in animals is that it helps to control the temperature, the body temperature. Okay, when you feel it too hot, go drink a lot of water. Water helps to regulate the body temperature. Water helps to regulate the body temperature. Other homeostasis. When you study homeostasis, you see how water is used to regulate the body temperature. Okay, this is in the case of animals. Now, how is water being used in the case of plant? In the case of plant, water is essential for one, photosynthesis. You know, what it is water plus carbon dioxide. When you add carbon dioxide to water, you get glucose. You manufacture glucose. So photosynthesis, the water is absorbed from the root hair cells. Water is absorbed from the root hair cells and is carried in the xylem. And then it take it to all the leaves where photosynthesis take place. Take it to the leaf where photosynthesis take place. Right. So water is essential ingredient or requirement for photosynthesis. That's number one in plant. Number two is that water is also needed to keep the plant cells turgid. You've heard of turgidity. If you want the plant cells to swell up, to get bigger, water is always needed, okay, to keep this plant cell moist and turgid. Is that okay? Number three is that water is used to transport mineral ions from the roots to the leaves of the plant. From the root, the root hair cells help to absorb the mineral ions, right? We've talked about this when we studied diffusions and osmosis and active transport. We talked about this. Now, the mineral ions that are being that diffused or that enter through active transport and diffusion or osmosis, when they get into the roots, they be transported. They added to the xylems, and then they mix with water. Water helps to move them up to the leaves for photosynthesis to take place. Is that okay? The water also responsible not only to transport the mineral ions from the roots to the leaves. The manufactured glucose from the leaf is taken to the rest part of the body of the plant with the help of water. The manufactured glucose is taken to the rest of the body of plants with the help of water. So water does two things here, like two interchange, like interchange station. Water carries the mineral ions that helps to manufacture the glucose. Then when the glucose is manufactured, water also takes the glucose to the rest of the body of the cells of the plant for usage. Is that okay? So water is very essential. Without water, then we're in problem. Both the animals and the plants are in problem. There will be no advantage of all the items that we have itemized, we mentioned. Now, why do we need the food? Why do we have to eat food? Why you can see this baby is eating food so that it can grow, it can become healthy, right? The baby can be healthy, and it can also have energy to play around. Because if you don't eat food, you don't have energy. You'll be feeling weak. You'll be feeling tired and weak and dizzy. And, you'll be feeling very light and weak and dizzy and dying. So we need food to keep going. The system, the human body, the plant, they all need food. Without food, we cannot grow and we cannot nourish and we cannot carry out activities and we cannot produce cytoplasm in our body that ensures that we are all right. Now, food is needed to provide us with energy. That's number one. Food is needed to provide us with energy. Remember the energy we have, we used to do so many things to do work. We use energy to work in the farm. We use energy to teach in schools. We use energy to carry out operations and treatment in hospitals. We use energies to do all kinds of jobs, to drive. We use energy to run, to sit, to everything you need energy. So food substances are needed to make cytoplasm as well. So this is the second functionality of food. Another one is that food helps to stay healthy. Now let's move on quickly. So if they ask you why are food important to us, we have to stay healthy, we need to produce more cytoplasm so that we can grow, and we also need food so that we can have energy to do activities. Okay? They will also ask you to discuss the roles of water in living things, to describe what we have talked about. Now let's go still on the biological molecule. After we've talked about water, the next one we're going to look at is called the carbohydrates. The next one we're going to look at is the carbohydrate. Now let's quickly take a look at the carbohydrate. Carbohydrate contains majorly three elements, carbon. Remember, water does not contain carbon. Contain carbon, hydrogen, and oxygen. Carbohydrate contain carbon, these three elements, hydrogen and oxygen. If it's simple glucose, we have 6,6, H12, and O6. Okay, this simple glucose, like the simple sugar. Simple sugar, which is called glucose, is just this molecule that we represent. Now, is that okay? So now we say carbohydrates, they are organic compounds because they already manufacture food. That's why they are organic. The mineral ions, they are inorganic. The mineral ions that is taken from the soil to manufacture food, they are inorganic substance. But the carbohydrate is already a manufactured food by the plant. So they are called organic compounds. So carbohydrates, if you want to explain or define it, they are organic compounds that are made up of carbon, hydrogen, and oxygen. Okay? The hydrogen and oxygen, they are in ratio 2:1. Or if you even include the carbon, you say ratio 1:2:1. Is that okay? So, but if you're looking at the hydrogen and oxygen, they're in ratio 2 to 1. Because 12 to 6 is ratio 2 to 1 if you break it down to the smallest ratio. Carbohydrates, which can be classified into 3 groups actually. Carbohydrate can be put into 3 broad groups. The first group is called the simple sugar, like the glucose and the fructose. The simple sugar, I'm going to show it in the next page properly. The simple sugar, which are the glucose and the fructose, and the double sugar, what we call the disaccharides, includes the sucrose and the maltose. Is that okay? The disaccharides. We call that double sugar, but the complex name is called disaccharide. This one, simple sugars, they are called monosaccharides. Both monosaccharide and disaccharide, they're tested using what we call the Benedict solution. We're going to talk about their test later on. We use the Benedict solution to test them, the presence of this simple and double sugar. We use the Benedict solution. The third sugar is called the polysaccharide. They are called the complex carbohydrate, the polysaccharide. I'm going to show them properly in the next page. Okay, they are called the disaccharides. The complex sugar are called the disaccharides. They are made from glucose. Many glucose come together to form a disaccharide. Is that okay? And this one, we don't test them with the Benedict solution. We test them with the iodine solution. So complex sugar, we use iodine solution to test for them. And simple sugar, which includes both simple and double sugar, we use what we call the Benedict solution to test for them. Remember this. Is that okay? Now let's look at carbohydrate properly, like I said. Now carbohydrates are organic compounds that are made up of 3 elements. The carbon, they are made up of carbon. They are organic compound. If you are explaining in the exam, they are organic compound or organic molecule. You can use molecules, you can use compounds. They are organic compounds or organic molecules that are made up of carbon, hydrogen, and oxygen. Now the carbon, the hydrogen, and the oxygen, they are in the ratio of 2 to 1. Why are they in ratio 2 to 1? Because it's 12 to 6. So hydrogen and oxygen are in ratio 2 to 1 because it's 12 to 6. So if I divide this one by 6, I'll get 2. If I divide this one by 6, I'll get 1. So that's why we say they are in ratio 2 to 1. That's why we say the hydrogen and the oxygen are in ratio 2 to 1. Just like water. Water is also H2O. So which is also ratio 2 to 1 in the case of water. So what we are saying is that carbohydrate, the hydrogen and oxygen are also in this format. Ratio 2 to 1. Remember this is ratio 2 to 1. They are also in this ratio 2 to 1. That's what we mean. Okay? Now let's move on. We say carbohydrates are divided into 3. Or you can say they are divided into 2. Now they are divided into what we call sugar and complex carbohydrate. Number 1, sugars. You hear sugar. When you have too much sugar in your blood, we say you have diabetes. If you have too much sugar is too much in your blood, insulins are not regulating the sugar in your blood properly, then the person has diabetes. And if you don't have enough sugar, also there's a problem, right? There's a problem. Okay? So both is not good. So you need to moderate your food. Not too much food, not too less food. Okay? So sugar, carbohydrates are divided into sugar and complex carbohydrate. 2 groups, sugar and complex carbohydrate, called the polysaccharide. Now these sugars are divided into 2. You have the simple sugar, which are called monosaccharides. The simple sugar, which we call the monosaccharide. Simple sugar, example of sugars, of course, glucose and fructose. Please don't forget, glucose has a hexagonal shape. I'm going to show the shape. The glucose has a hexagonal shape, while the fructose has pentagonal shape. The fructose has a pentagonal shape. Now for double sugar, they are called disaccharides. For double sugar, they are called what? Disaccharide. Disaccharides includes maltose and sucrose. Maltose is 2 glucose joined together. Okay? If a disaccharide, di means 2. You take 2 glucose, you add them together, you get maltose. You take 1 glucose and 1 fructose, you add them together, you get sucrose. This one is not 2 fructose. It's 1 glucose and 1 fructose. You add them together, you get sucrose. Is that okay? So these are disaccharides. which are called double sugar. These are monosaccharides, which are called simple sugar. The third one are called the complex carbohydrate. They include number one, starch. You'll be hearing of starch, like bread, like cereals, like there's this popular one, the one Quaker oat, like the oatmeal. They are all complex sugar. You have cellulose, like some grasses and so on and so forth. You have glycogen. Okay, these three are good examples of complex carbohydrate, and they are called polysaccharide. Now let's go to the next one where we explain them in details. Now we start with the simple sugar, the single sugar, right? Sugars are divided into single one and double ones, right? You know, right? So we start with the single ones. Single ones are glucose and fructose. You know that, right? Now the glucose has what? A hexagonal shape, 6-sided. A 6-sided polygon, that's a glucose. A 6-sided polygon, that's a glucose. A fructose is a pentagon, a 5. 1, 2, 3, 4, 5. 5-sided shape is a fructose. Now, simple sugars cannot be further digested when you eat food. When you break food down, when you break carbohydrate down in your body, then they reduce to the simplest sugar. So they are the simplest unit of carbohydrate. They are the simplest unit of carbohydrate. They cannot be simplified further into smaller unit. This is the smallest unit. Okay? They can pass through the cell membranes and they can be absorbed into any cells. Now, glucose and fructose, they are common examples of fructose sugar. Now, let's move to the double sugar. Double sugars. You know, we say sugar are divided into two, single sugar and double sugar. The single sugars, they are called monosaccharides. The double sugars are called disaccharides. Now, double sugar is nothing other than you just take two simple sugar, add them together, you get a double sugar. For example, if you react a glucose with C6H12O6 plus another C6H12O6, react them together, they form a maltose. They form maltose. Two glucose react together, they form maltose and water. Maltose. So this is how maltose is made. Reacting two glucose together, you get two glucose molecules joined together with a link or a chain. So this is what we call a maltose. And the test is also Benedict solution. The test for both simple sugar and double sugar is Benedict solution. Okay, we're going to talk about the test. Okay, now what is the case of sucrose, which is also a double sugar? Sucrose, like sugar cane, a good example is sugar cane. Okay, a good example of sucrose is sugar cane. And sugar cane, not only sugar cane, carrot also. Carrot that we eat, or sweet fruit, any fruit that is very sweet. Any sweet fruit you eat, they are sucrose. We call them sucrose. They are double sugar. They are just double sugar. Okay? Sugar cane stems, like these ones, they are double sugar. They are made up of one glucose and one fructose. One glucose and one fructose. You get sucrose. Now, let's move on quickly. How do we test for reduced sugar? Reduced sugar comprises of both the simple sugar and the double sugars. So we call them, how do we test for them? We use what we call the Benedict solution. We take a solution called the Benedict solution and we add it to the sugar. And then when the solution remains blue, we say nothing has happened, there's no sugar. Okay? You add the Benedict solution to any simple sugar because the Benedict solution itself is blue. When you add it to any sugar, like sugar cane, you add this Benedict solution to a sugar cane. Sugar cane, I mean the water inside, not the body. I'm not talking about the body. I'm not talking about when you peel this body, the inside, the one you chew and where you get the sugarish water from. Okay? So when you add this Benedict solution, this is a Benedict solution to it, and it remains blue, it means there is no sugar in that substance. When it remains blue, when the blue solution remains, it means there is no sugar in that substance. So we use Benedict solution to test for both glucose, fructose. These two are simple sugar, galactose, maltose, and lactose. These are double sugars. These are double sugar and these are simple sugar. So any of these we can add the Benedict solution to it. Okay, so when you add the Benedict solution to any of them and the solution, because the solution is blue, and if you add it to any of these, the glucose, any glucose substance, or any fructose substance, or galactose substance, or maltose substance, or lactose substance, if you add the Benedict and it remains blue, it means there is no reduced sugar. It means there is no reduced sugar in that substance. But if you add the Benedict solution and you see that the color changes to green, green precipitate is formed, then there is small amount of, if you find green color, there is small amount of reduced sugar. There is small amount of reduced sugar, traces of reduced sugar. The reduced sugar is not a lot. There are just little traces of reduced sugar. Now, if the blue color, if you add a Benedict solution to any of these, and the blue color turns to yellow or orange, like this one, like this number 3 here, the blue color turns to orange, it means that there is moderate amount of reduced sugar in that substance. In that your sugar cane, or in that your any maltose or sugar cane, any of the substance you are testing for, that is either simple or double sugar, single or double sugar. If you have yellow color or orange color, or a mixture of both, then there is moderate amount of simple sugar. But if you add a Benedict solution, remember the Benedict solution is blue, and the blue changes to brick red precipitate, then we say there is large amount of reduced sugar, like this one. If it changes to a brick red, we say there is large amount of reduced sugar. There is large amount of reduced sugar. So these are the 4 options you find in multiple choice exam when you test for a simple sugar. When the outcome remain blue, if you add a Benedict solution to any simple sugar, okay, to any sugar, then and it remain blue, that means there is no reduced sugar in that substance you tested for. If it changes to green, there is just small traces of it. If it changes to orange or yellow, then there is a moderate amount of reduced sugar. If it turns to red, brick red, there is large amount of reduced sugar. Okay, so that's how we test. Now let's go to the next one, the third one, which is called the polysaccharide, the complex carbohydrate. A good example is the bread, the wheat bread you buy, you eat, the flour, the potato. They are all complex carbohydrate. When you eat potato food, it's a complex carbohydrate. But the good news about potato, it contains a little bit of other nutrients. It contains little vitamins, it contains little protein, contains little of other. But it's mostly carbohydrate. It's mostly carbohydrate, but it contains little of every other food nutrient. Okay. Now, if you take oatmeal, pasta, macaroni, all this type of food, Nestor, you just measure by a few. They are called the complex sugar. Now, the complex sugar is different from many monosaccharide or simple sugar that are added together. When many simple sugar add together, you add them together, you get a polysaccharide, you get a complex sugar, which is called starch. A good example of a complex carbohydrate is starch, glycogen, and cellulose. So a good example of complex carbohydrate is what? Starch, glycogen, and cellulose. Okay, now let's move on. We say we can test how to test for starch. We use the iodine solution. We don't use the Benedict, please. Don't say, we use Benedict to test for both the single sugar and double sugars, so we can also use Benedict. No, Benedict solution is only for reduced sugar. Reduced sugar comprises of simple sugar and double sugar. Okay? Now, for complex carbohydrate, we use the iodine solution. Now, you take the iodine solution, just add it to a potato. You peel a potato, you just add a drop of iodine to it, to the potato. Okay? Iodine solution, when it turns a blue-black with the potato, then it means there is a complex carbohydrate in that substance. If it turns blue-black. Is that okay? This is how we test for complex carbohydrate. Now let's take a look at the functions of carbohydrate. What do we use carbohydrate for? Number one, carbohydrate is used as a source and store of energy. We use it as source of energy, and we also use it as store of energy, carbohydrate. Okay? It's for energy. It's about energy. Gives energy. Number two, it forms a supportive structure, such as your cellular cell walls in plants. The cellular cell wall in plant, that supporting rigid structure that give the plant rigidity, is formed by complex carbohydrate. Number 3 function of carbohydrate is that it's used for the synthesis of nucleic acid, the formation of nucleic acid. Number 4 is that it's used to make lubricants like the mucus. It's used to make carbohydrates is used to make lubricants such as the mucus, which cover the inner surface of the trachea. You know the trachea in the respiratory system. It covers the inner surface of the trachea and traps the dust particle, helps to trap the dust particle. So carbohydrates helps us to provide or produce mucus. Now also to make nectar in plant, that sweet smelly substance that attracts insects. Okay, that attract insects. Carbohydrate is used to produce that sweet smelly substance in plant. So you pause the video, like and subscribe and share, and practice these questions to see whether you understand the part we have done. How can you identify reducing sugar? How can you identify starch? To identify this, you have to use what? The Benedict solution to test for it. And to identify this, you have to use the IOD solution to test for it. Okay? Outline the roles of carbohydrates in the life of living organism, which we just mentioned. Store of energy, use of energy, store of energy, production of mucus, production of sweet smelling substance in plants, and so on and so forth. This helps in the production of cellular cell walls, and so on and so forth. So, yeah, you should be able to answer all of this. Now, let's go to the third one, which is the fat. We've studied water, we studied carbohydrate. Now we are in the third biological molecules. Number 3 part of the biological molecule is called the fat. Now, what are fats? You often call it fat and oil. So sometimes you see it goes together, fat and oil is same thing, okay? Whether we say fat or we say fat and oil, we're talking about the same thing. Now, fats are organic compounds or organic molecules that are made up of carbohydrates, that are made up of carbon, hydrogen, and oxygen, but contain much less oxygen in production. So, it's almost the same makeup, but the only difference is that the oxygen is way lesser. Okay, the oxygen is way less. You know, in the carbohydrate we write simple sugar, we write 6, 12, 6. The oxygen may be 5 or 4 or 3. It's lesser in this regard. Okay, now these fats can be identified using what we call the ethanol emulsion test. We use the ethanol emulsion test to carry out the fat identification of fat in foods. Now, how the outcome is that it turns cloudy in the presence of fat. So when you add an ethanol into a fatty food, it changes the color to cloud color or milky color. Okay, we're going to come to the test classification. But let's take a look at what are fats. Let's quickly look at the meaning and the formation of fats. Fats are organic molecules, like we said already, that are made up of carbon, hydrogen, and oxygen, but even though the oxygen is way less. Unlike carbohydrate, fats, they contain much less oxygen in proportion to hydrogen. So the ratio is not ratio 2 to 1. It's not ratio 2 to 1 because the oxygen to hydrogen is way less. Now, fat molecules, they are made up of 1 molecule of glycerols and 3 molecules of fatty acid. Every fat molecule, if you take one fat molecule, like this one, is made up of 1 glycerol, like represented with this red, 1 glycerol with 3 fatty acids. These ones are fatty acids, these are the fatty acids. So this is a fat molecule. So when you digest it, during digestion, this will break into the fatty acid and the glycerol. So you have 3 fatty acids reacting with 1 glycerol to form a fat. Okay. A good example is your cooking oil, your margarine, your cheese, your butter. These are examples of food where you can get fat or oil. Okay. So when you eat use cooking oil to fry your egg, you use margarine, butter, margarine, cheese, hawa, the Arab one. Okay, you have hawa, Hawani or Ha. Okay, anyway, you know what I'm talking about. So when you all of this food they give fat. Okay, they give fat. Now let's take a look at the food. It includes butter, cheese, which I've already mentioned, fatty meat. You know meat, the fatty part. You know meat also has the fatty part. When you eat the fatty meat, you get fat. Olive, when you take olive oil or you take the olive itself, okay, you get fat. If you take nuts, although nuts give protein, but they also give fat. If you take many nuts, you get fat. Peas, they give protein, they give fat. Beans also, they give protein, they give fat. Seeds, they give protein, they give fat. And castor oil and palm oil. The palm oil is very common in Africa. The palm oil, they have a lot of palm. Malaysia also has a lot of palm. Palm oil and castor oil, they all give fat. Okay, let's move to the next one. How do we test for fat? We say use the ethanol emulsion test. So remember the reducing sugar. Reducing sugar comprises of two types of sugar, the simple sugar and double sugar. But that reducing sugar, we use the Benedict solution to test for it. And we say if you add, the Benedict solution is blue solution. If you add the blue solution to it and it remains blue, that means that substance do not have a reducing sugar. If it turns green, it has only traces of reducing sugar. If it turns orange or yellow, it has moderate reducing sugar. If it turns brick red, it means it has a very large amount of reducing sugar. So that's how we dealt with simple sugar or reducing sugar. Now, when we came to polysaccharide, which are complex carbohydrates. We say you use the iodine solution. You add it to it. If it turns blue-black, then it means that the food substance contains iodine, contains carbohydrate, complex carbohydrate. Then, now we are in fat. To test for fat, you add the emulsion. We use the ethanol emulsion test. Okay. Now the ethanol, the color is like color of water. Look very similar to water. So the ethanol emulsion test is a test for the presence of fat. Now when the substance, the food now turns cloudy, a cloudy white emulsion is formed when the water is added to okay, we say a cloudy white emulsion is formed when water is added to the mixture of the ethanol and coconut oil. When it's added to ethanol and coconut oil. Okay, sorry for that. We're trying to. Now we say the ethanol emulsion test it helps us to identify fat. And then whenever it's added to a fatty food, a cloudy white emulsion is formed. And when a cloudy white emulsion is formed, it means that the water is added to a mixture of ethanol and coconut oil. So you take coconut oil. Coconut oil is the fat. You know it's the fat, right? It's the fatty food. Palm oil, coconut oil, you want to test. You take the coconut oil, you add ethanol to it, right? You also add water. And then, when the mixture begin to change cloudy, you know the color of water is not cloudy. It's a bit settled. Water is settled. You take a test tube. You add all three in a test tube. When the mixture is changing to a cloudy or whitey or something like that, then it means that the coconut oil is the fat food that you're testing. You can take palm oil. You can take cheese and add here. Okay, you add water, mix it with the cheese so that it will become a bit liquid, and then you add the ethanol. Okay, ethanol. You add the ethanol. When it start gradually changing cloudy, then we say when a cloudy white emulsion is formed, then we say that the food contains fat. So a cloudy white emulsion is also formed when water is added to ethanol that has been mixed up with peanuts. You know, many peanuts, they contain fat, okay, which indicates the presence of fat in peanuts. So now let's go to the functions of fat. Because this one is easy, we say take coconut oil, take water, take ethanol, add them together in a test tube, shake them, leave them. After a while, it turns cloudy. Once it turns cloudy, it shows that there is presence of fat in the food substance. Okay, in the coconut oil. Now let's go to the next one, the functions of fat. What fat does to our body. One of it is that it's used as a source and storage of energy. Fat is used as source of energy for your body and also store of energy for your body. Okay, it stores energy for you. It's used as storage. Okay. Number 2 is that it's used as an insulating material to prevent heat loss. To prevent heat loss. The fat in your body helps to block the loss of heat in your body. So they are used as insulating material. That's why people who are very fat or chubby, they can withstand cold more than people who are slimmer. So when you have more fat in your body, you are able to withstand the cold. You are able to withstand cold more than a slim person, because it will help to prevent heat loss in your body. Okay? Number 3 is that fat is used as a solvent for fat-soluble vitamins and hormones. They are used as solvent for fat-soluble vitamins and hormones. Solvent means something that can help to dissolve other substances. Okay? Now, we say another function of fat is that it's used as essential part of the cell membrane. It consists or comprises of the cell membrane you hear in our body. And then another one, it helps to reduce water loss from the skin surface. That's why someone who is fatty doesn't easily lose water. There's no easy excess of water lost in the body. Or like slim person. When slim person goes into the sun, easily can lose, when it's heated up, easily can lose water. So you can pause the video, like, share, and subscribe, and answer these questions. Is that okay? Answer the question. Now let's move on to the third one, which is the protein. Now the protein is the 4th part of our biological molecules. Remember we studied water, carbohydrate, fat. Now the 4th one is proteins. Now proteins are organic compounds that are made up of additional to the first 3 things we know. You know, we used to say carbon, hydrogen. I'm using the symbol, I'm just writing the symbol. Carbon, hydrogen, and oxygen, right? In the case of protein, there's additional elements like nitrogen. There's even sometimes additional element called the sulfur. So, but most proteins, they just have these 4. But some proteins have this additional sulfur. Okay? So we say proteins are organic compounds that are made up of carbon, hydrogen, oxygen, and nitrogen. Or even in some cases we add sulfur, right? Now protein consists of amino acid. The smallest unit of protein is called amino acid. Amino acid, the smallest unit. Just like the smallest unit of carbohydrate is what? Glucose. The smallest unit of carbohydrate is glucose. The smallest unit of protein is amino acids. Many amino acids alter themselves to form what? To form protein. The smallest use of fat is glycerol and fatty acid. Smallest unit for fat is glycerol and fatty acid. Okay, so amino acids alter themselves to form a chain, a complex chain called the protein. Proteins can be identified using what we call the Burette test. Please remember. Now we've mentioned about the Benedict test for simple sugar. We mentioned about the iodine test for complex sugar or complex carbohydrates. We've mentioned about ethanol emulsion test for fat and oil. Now we are mentioning the Burette test for protein. So you must know all the tests and the result, the color change. You must what? Know all the result and the color change. Right? Now, in the Burette test, when the blue color remains, then it indicates the presence of protein. It remains blue in the presence of protein, right? If it turns violet in the presence of, remains blue in the presence of protein, turns violet in the presence of protein. Both. Now, let's see how. We have a test for it. First, before we look at the test, let's look at protein. Protein, the amino acid, the good thing about amino acids is that they are all different. Each amino acid, some are circular, some are rectangular, some are triangular, some are hexagonal, or tetrahedron, some are quadrilateral, some are circular, some all different shapes. So all these different shapes are called, each one is called amino acid. Amino acids are different. They're not all same. The amino acids are all different. So when they're linked together with a chain, they become a protein. Now proteins are organic molecules or compounds that we said they're made up of carbon, element, hydrogen, oxygen, and nitrogen. Sulfur may also be part of protein in some cases. Proteins, they're present in all cells of human body or of animals. Proteins are present. If there's no protein, then you don't have enzymes. You know, enzymes are proteins. Proteins are present in all cells and always present for metabolic activities. Protein molecule is built up from simple compounds known as the amino acid, which we already explained. Amino acid add onto itself, and amino acids are all different shape and sizes. They add onto itself to form a chain, which is called the protein. Now let's move on to what are proteins. We say enzymes and antibodies. You know the antibodies? They are made up of proteins. Enzymes and antibodies are made up of proteins. So you see why you have to eat a lot of proteins? So that your body can produce enzymes, your body can produce antibodies. Antibodies help to fight against diseases. So these are proteins. If you are eating carbohydrate, only rice, bread, rice, you are not giving yourself the adequate antibodies or the adequate enzymes and the adequate substances that helps you to grow better. Now, each protein has its own unique three-dimensional shape that enables it to carry out its function. Each protein has its own 3D shapes, quite different from any other protein. Protein are not all same. Food rich in protein include milk, so you should take a lot of milk, eggs, a lot of eggs, seafood. Many people don't like seafood. They contain a lot of protein. Then the meat itself contains a lot of protein. Soya beans contain a lot of protein. Nuts like groundnut, peanut, the one you call peanut, hazelnut, almond nuts, cashew nuts, all forms of nuts, they contain protein. Grains, they also contain small amount of protein. Grains, like the maize, the corns, they all contain small wheat. They all contain small amount of proteins. Okay? And then vegetables. Okay, vegetables not only contain vitamins, they also contain small amount of proteins. And also what we call the French beans, they contain small amount of proteins. So these are foods like meat. You can see different meat and fish that are caught, okay, which contain proteins. Okay? And the enzymes of protein includes the Pepsi and trypsin. Okay, they include Pepsi and trypsin. This is how the molecules are in the body. Okay, the site for where protein molecule is attached. This place. Okay, this is site where virus or any other disease is attached in the body. So an antibody helps to. This is the structure of an antibody in your body, which are made from protein. Antibodies, they help to fight diseases, fight viruses and bacteria in your body. So if you're not eating a lot of protein food, you'll not be able to produce antibodies like this to fight bacteria and virus. Okay? Then enzymes. If you're not eating a lot of protein, you don't have enzymes that will help to digest your food. You know, enzymes help to digest food. They help to speed up, break food molecules into smaller units. Is that okay? Now, how can we test for protein? We say you use the Burette reagent. Proteins can be detected using the Burette reagent solution, right? A violet or deep purple, okay, a violet or deep purple color is observed when the egg white solution is mixed with the Burette reagent. This indicates the presence of protein. So when you take a Burette reagent solution and you add it to egg white, okay, and you get a deep color, a deep blue or a violet, not really deep blue. You get a violet or deep purple color, then it means the food contains protein. In the absence of protein, the solution will remain just blue. So if the solution remains this blue, it means that there is no protein in that food. Because the reagent color is blue. This reagent is blue. So when it remains blue, it means that food there is no protein. So you can actually test for protein. You can go to the market chemical shop to buy burette solution. Add it to the food and see whether it returns to a deep purple or a violet solution color. When it does that, then that food you're about to consume contains protein. Now, what are the functions of protein? Oh, protein is super important. Remember that. They're used to make new cytoplasm in the cells. Proteins are used to make what? Cytoplasm. You must remember this. Number one. Number two, they are used to make what? Enzymes and hormones. So if you're not consuming enough proteins, you will not have many digestive enzymes to break down your food. So you'll be having constipation, indigestion, and so many problems. And you may have the adequate hormones to regulate your mood and to keep you in a better form. Okay, so you need proteins. Proteins are used for the formation of antibodies, we already mentioned this, to fight against diseases or pathogens in the body. So we mentioned the manufacture of cytoplasm, the manufacture of enzymes and hormones, the manufacture of antibodies to fight diseases, and also proteins are used to make chromatin in chromosomes. They are used to make chromatins in chromosomes. So if you don't have enough protein in your diet, you suffer a lot of things. So here are some practice questions. Pause the video, like, share, and subscribe, and answer these questions to see whether you're following or you're getting it or you're far from it. Now we go to the last part of today's class, which is called the DNA. The full meaning of DNA, many people don't know. They just hear DNA. You know, DNA is used to check whether you are your father's son or a father's daughter. If they want to find out whether you are a child of your father, they will go and carry out what they call the DNA test. Okay, to confirm whether you and your father have same DNA. If you have 98% match, then you are your father's child. If you do not have up to 90-something% match, then you are Yugoshoko. Okay? So the full meaning of DNA, it means deoxyribonucleic acid. Okay, the D is up to this place, the D. Okay? Dio. Like oxyribo. So deoxyribonucleic. This nucleic, N is the nucleic. This N is the nucleic. The deoxyribo is the D. Nucleic is the N. And the acid is the A. Okay? So DNA means deoxyribonucleic acid. Okay? Depends however you pronounce it. Okay? So you know a DNA. Now what is DNA? This is the last part of our biological molecule class. Okay? Now a DNA carries the genetic information for the development and the functions of the body. The genetic information, what you are made up of. Okay, the code, the genetic information that defines who you are, how you look, your behavior, and all of these things. You know, we say there are two things that affect how you turn out. The genetic factor, which is your DNA, and your environmental factors. Okay? So the DNA, first of all, comprises whether you be brilliant. If you're born from brilliant parents, you definitely carry a DNA that is supposed to make you brilliant. The other one is the environment. You may be from a brilliant parent, but you are in a very low environment. You take it to the bush, in the jungle. You never learn anything in your life. You just run and follow the animals in the bush. And then you will ultimately not be smart. Okay, that's on the light side. Now let's move on quickly. So we say DNA carries the genetic information of who we are, the code of who we are. Okay? Then we also say that the DNA each strand of the DNA consists of a sequence of bases. Every strand, this is a strand, this one. This is a strand, this is another strand. Every strand consists of sequence of bases. Base, base. We mentioned there are 4 bases in the DNA. One is called C, G, A, and T. Okay, we're going to talk about that. There are 4 types of bases in a DNA. We have base T, base C, base G. Now, A we always pair with T. We'll show you why. A we always pair with T, and C we always pair with G. We'll show you. Okay, now let's begin to look at it from the study perspective. Okay, let's look at it from the study perspective. DNA, which is a deoxyribonucleic acid, carries the genes or the genetic informations that is necessary for the development of human beings or of any organisms, and also carries the functions of the body of that organism. The functional code, the code formula, the algorithm, the coding that has been done by your creator is the DNA every code is embedded into. Okay, is the code, the algorithm, the functionality of the organism are coded in a DNA. Okay, now a DNA molecule has a double helix structure. Now what do we mean by helix? The twisting. This is one helix. Double helix is like you twisted them. Because DNA is, for the purpose of study, yes, we look at it like a ladder. Like a ladder. But DNAs, they are not like ladder. They're helix. They're twisted like this. Okay? Okay, so this is DNA, how it looks. Here is a base of a DNA. Here is another base. This is one base, this is another base. Okay? They link. This is A and T. You see, they fit into each other like enzymes are substrate. Like enzymes are substrate. This is C and G. This is for C and G. They are like an arc, length of an arc. They fit into each other. Okay, this one is like a pointed mouth, like a nail. They fit into each other like enzymes are substrate. Okay, we're going to look at this now. This is how they look. This is called a part of a DNA molecule, and which are usually a double helix structure. Okay, in the exam, this is how it will be drawn and shown to you, like this one, because this is the practical look of it. Okay, now let's take a look at, we say there are 4 types of bases in a DNA. We have base A, base C, base G, and base T. And we say A we always pair with T, remember that. In multiple choice, you find this a lot. A we pair with T, and C we always pair with G. Now look at C and G. C looks like this, and G looks like this. That's why they pair, because this can fit into this, and this sharp mouth can fit into this active site, like active site. This is called the base pairing of the DNA, the base pairing of the DNA, the DNA strand. Now let's move to the next one. What is a DNA? What is a DNA? Now, each species of organism has a unique sequence of bases in DNA, right? Unique, each one. Every organism do not have exactly same. Each one has a unique one. If you take a look at this A, B, look at this one, is not same as the second one. If you take a look at the third one, it's not the same. So each one is kind of different. For this A, B, C, right? Each one is kind of different. Now when we take the B and A, we mix it and heat it, we will get this 8 base pair form. We get this one. We get this one. You mix this A and B and you will get this. You mix B and C, you heat them, you get this. So this is the final stage of a DNA, because they don't all look same color. Okay, they don't all same color. The pairings are not all same color. Okay, because if you say like this, does it depict A and T now? But now, right now, yes. Okay, so this is how the end product, before base pair are formed. This is for the A, this is for the 4 base pairing of a DNA. Okay, let's go to the next one. Here you have, okay, you have some question, you pause, you like, you share this video, and answer this question. What does the DNA stands for? Remember, D, also ribonucleic acid. So you should be able to tell what it stands for. Name the bases that makes up a DNA molecule. There are four bases that makes up A, T, C, and G. So you should be able to answer these questions. Here is a summary of what we've talked about, which you can fill it up yourself. Okay, fill up this summary. You give example of double sugar. Sucrose is a double sugar. Single sugar, you can say glucose or fructose. Here is maltose and sucrose. So you fill it up. Okay? The test, you use a Benedict solution to test for reduced sugar. When the color is blue, it means there is no reduced sugar. When it's orange, it means there's moderate. When it's brick red, that means a lot, and so on and so forth. So you fill up this one as something like a note, a small summary of note. Pause the video and fill up this one as a summary of what you have learned today. And the fill is here, but you do it yourself first before you. So with this, we come to the end of our today's class. Thank you for joining us.